Basically going to get started. So first of all, before I start, does anybody have any questions or issues that they'd like to bring up?
Would you be able to tell us more about the written assignment that's required, please?
Sure. Okay. So first of all, a number of you, I think about at least 4 or 5 of you, have shared with me a number of the articles that they would like to go and do, which was fine. And obviously, you can talk among yourselves. So some of you who have gotten feedback from me and know what you're doing, you can share that with other people. So again, the nature of the writing is roughly a 5-page double-spaced short paper. So what you're basically doing here is, I would imagine the first half page is you're introducing the topic that you're interested in doing. And then the last half page, or let's say the first page and the last page, you're spending writing what you're doing, and then the last page is a sort of overall summary of where you think things are going. And the 3 middle pages can be devoted to the 5 articles, a little bit like a half a page each, of describing the important findings and maybe the differences in techniques that were done across the studies. So for a number of you, what you typically do is pick some type of either neurological or psychiatric disorder, and then what you may end up doing is looking at changes from either fMRI or SPECT or some type of anatomical technique that begins to correlate the symptomatology or certain types of issues with particular brain changes, which is all perfectly fine. Again, you can do any kind of paper that, as long as the dependent variable is a neuroanatomical measure that's included in there, that is the important thing. So again, let's face it, when you're reading, let's say, a 10-page paper, you're not going to spend a little bit more than half a page allotted to that paper with really in the weeds details. The only point in which you might want to go into the weeds in terms of a detail is if you read 2 papers that come out with literally diametrically opposed views. Some people see increases versus some people see decreases in, let's say, the same brain area. So what it's going to be important for you to do is understand why. Is it, this is how things happened? Let's face it, whenever we're doing studies and we use statistical significance, what you're basically doing is you're talking about a strength of an effect. So if the strength of the effect is P is less than 0.05, that means in 5% of the time, the results that person is seeing is due to chance. So maybe you picked out 2 studies that literally saw a chance and they were diametrically opposed. Sometimes when you have things like that, you then really have to go into the weeds to figure out, well, just because these people are saying there's this group and this group, maybe there's differences between the groups. and then you would write that, and that would then affect somehow your conclusions. So that's basically what I'm asking you to do. Is I'm asking you to exercise some degree of critical thinking in terms of your in terms of the papers. Is that clear? Okay.
Yeah, I think so. So just to summarize there, you're saying you need to do a 5-page double-spaced paper,
Yeah.
half to 1 page of an intro, half to 1 page of a summary at the end. The main body
and
is
then the middle is basically going into the specific results of each of the 5 studies that you selected.
You're interested more in the results than the methodology, but we can compare technique.
But again, the method, look, if everybody's using almost an identical methodology, so for instance, no one's done this yet, but you could pick 5 papers coming from one specific lab who's literally using the same methodology over and over and over again with either different independent variable manipulations. So all you'd have to do is describe that methodology once, and what you have there at least is the understanding that how whatever they're measuring in a very consistent way. Once you go across labs, just because lab X says I measure A and lab Y says I measure A, perhaps the differences in how they measure A are different, or the samples are different that you then end up recognizing or something. Okay, so it's up to you when you're reading those articles, especially when you start to see quite discrepant results between articles, to at least begin to try to figure out what the hell's going on here. Or is the field a bloody mess? I mean, very often you have this. Look, I remember when I became executive officer back in, I was named to that in the late spring of 2016. And my predecessor, who now is the president of the University of Palo Alto, which is a pure psychology graduate school for all these different things, she was having a research day. We have that every May. And what she had was a panel of people coming in talking about why you can't replicate results in psychology. That there were some studies done back around 2015 that found for particular fields of psychology, when people try to just do a direct replication of the results, they failed 40% of the time. So of course I look at this. I've been in the field since 1970. I published my first paper in 1973, and I'm going, I never heard of such a thing. So of course, when I looked into it a little bit further, what they were talking about is social personality psychology, which again is very, very much based on correlations and things like that. And of course, that might very much depend on whatever sample pool you're working with. Obviously, when we get into something like neuroscience, we hope that we have a greater degree of stringency. And again, I say that, I'm not saying that it's perfect, because as I pointed out during this course, I really try to give you somewhat of a historical vantage point. And, so the area that we're about to be in, and the area we just covered, the hypothalamus, in the 1950s and 60s, people were talking with certainty that they had discovered something, the feeding centers in the brain, blah, blah, blah. And it would be study after study after study. And then finally, when some discrepant results started to come out, it really, and obviously further information about those structures came out, it became abundantly clear that this was not a center at all. That, and I mean, that's what science basically goes. Let me just tell you something as to how I think I got influenced by this. I was a history major in college, and in my first couple of years, I think I majored in Barstool. How I'm in the position I'm in today is an absolute miracle. Where I basically say this, but in the when I decided, to leave the Air Force and go back into teaching, I thought I was going to go back and maybe become like a high school guidance counselor because the jobs weren't there. So again, I entered this master's program in psychology at City College, and then I started working in a lab, and I found where I wanted to go. But I'm going to basically tell you an absolute parallel happened to me with PBS, the public broadcasting system. In the around 1972 or 1973, they created probably now a very regrettably named named program called, very sexist, it was called The Ascent of Man. Okay, you can basically Google that, and it came out of a book, but then it was a 13-part series, basically moving all the way along of humankind. And what it was narrated by, very, very interestingly, was a guy by the name of Jacob Granowski, Bronowski, who was a Polish mathematician slash philosopher slash physicist. And he did work as part of the Manhattan Project. And this guy really, when he did it, he was probably in his mid 60s or late 60s. He had an incredibly rumpled appearance to him. He always had glasses. That what I have to always do is at the age of 74, go, I'm not seeing well with my glasses. I must be going blind. Then I take them off and I look, and of course my glasses are dirty. So I mean, he would always need a cleaning of his glasses, and he wore classic Eastern European, ill-fitting dark suits. But the man was an absolute genius in terms of presenting this stuff. And in the last episode was entitled Certainty. Okay. And of course, the point is, in science, what we are looking for, just like any other kinds of human beings, is we're looking for certainty. We want to know what is and how we can exist in a very crazy world. And the more certain we are, the better. And then he starts to talk about how certainty could basically affect things, especially when science and politics begin to mix. I don't have to tell you, we've just gone through, by the way, today is November 12th. March 12th was when I decamped from, I heard that Queens College was closed for the day, and I decamped from my house in Pleasantville and rode up here to join my wife up here in the Northwest Catskills. And I've left for only one day since. So we are in a world of the pandemic and whatever, and we want to know what's true. So what Granowski is doing is he's walking, and he's walking in the background is this absolutely bleak environment, very, very gray clouds and whatever. And what you saw behind him is a field, that it's now late fall or maybe very early spring, sort of things are dead around there. And he's walking through and talking about the search for certainty and people who believe they have certainty. And then you realize as he's walking, he's now stepping and you're hearing sloshing, and he's literally walking into a pond. And he basically goes, throughout history, we have found certain times when people have discovered certainty. They know the absolute truth and they acted on it. And this is where such an action went. And he bends down and he's standing, knee deep in this pond of muck and whatever. And he with his ill-fitting suit, reaches down and pulls up the muck and whatever. And he goes, here is what certainty is. And what is he standing in? He's standing in one of the ponds that would be found around Auschwitz, where people were exterminated. And they were exterminated because the people who were doing it were certain. Okay, so I mean that I know I'm going back to a 1960s phrase that blew my mind, and then I really felt in a very, very deep sense what enterprise I was doing and studying. That what we don't have all of the facts. We live in a probabilistic universe. Shit can happen and things like that. And so I view doing this sort of little exercise that you're doing in a way, to enter yourself. For some of you, you're already ensconced in the scientific field with a series of labs and everything. But I always feel like this is just a very good exercise. Plus the practical exercise of being asked if you want to do something and then apply for funds to do it. And then, but they gave you that there's an RFP, a request for proposal, and you sit there and you write this magnificent proposal, but the proposal has nothing to do with what they want. So sometimes people ask you for things that they want, and that's why I basically go and do this. So please don't overthink this or whatever. I'm just going back to that detective and dragnet. Just the facts, ma'am. Just the facts. Lay out what you. And again, I go back to my time in the Air Force as an intelligence officer, where I had to get up and give a 2-minute briefing. And the way the Army or the military gives briefing is what? Tell them what you're going to tell them. That's that first page. Tell them, give the results and any changes in methodology in the middle, and then tell them what you told them, which is that last page. And in so many ways, that is the easiest kind of way of doing it. And if you read many papers, you sit there in a discussion and you'll have some people writing that this was the greatest study of all time and blah, 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 and whatever. I bring you to another paper, exactly the last sentence of another paper written in 1954. The last sentence, I'm going to paraphrase it. I don't have the exact sentence, but the last sentence of that paper basically said, these results may have implications for the further understanding of biology. And who were the authors of that paper? James Watson and Francis Crick, when they basically described the DNA helix. Okay, here they had probably the most monumental discovery of the 20th century. I know there's others in other fields and whatever, but you have one of the top 10, and they wrote a discussion at the end that might have implications for. So, this is how we tend to couch things. This is why people in science are utterly ridiculed, because let's face it, about 330 million folks who live in the United States, at this point, they crave certainty. And sometimes some people go one way, or sometimes some people go another way, because, by the way, those two other ways of saying things in some way, shape, or form bring you a promise, the specter of certainty. Okay? So I hope I didn't spend too much time on that. There we go. All right. So now we go back to whoop, sorry, wrong one. Okay. I have too many files sitting here. There we are. Okay. Okay, so we basically started on the limbic system last class, and what we were moving first into is one of a very important limbic system structure called the amygdala. So first of all, I'm assuming right now, John, that you're recording. Okay, great. Okay, so when we look at the amygdala and we talk about the amygdala nucleus, they are actually a group of nuclei. So what you basically see is very often three general areas called the corticomedial nuclei of the amygdala, which are primarily involved in receiving information that is olfactory. Then we have the basolateral nuclei that are receiving enormous numbers of inputs from many of these other areas that you see here on the left, the hippocampus, the sensory cortex, the thalamus, the septum, the prefrontal cortex, hypothalamus, and brainstem. And they sort of filtered through the basolateral nuclei. And then the basolateral nuclei then synapse into the central nuclei. And then the central nuclei, in turn, is the major output of this amygdaloid complex. So here we're basically seeing a lot of so things like the sensory thalamus and sensory cortex come through the lateral nucleus into the basolateral nucleus, and the basolateral nucleus can project to the dorsomedial nucleus of thalamus and ventral striatum, but and also there are inputs into the basal nucleus. So what you're doing is getting a big feedback into the central nucleus. And then the central nucleus, as you can see, is the major output, going to the hypothalamus, going to the midbrain, going to the pons, and going to the medulla. But So one interesting thing, again, to begin to try to tie together a number of the structures that we basically are that have talked about, is we recognized that when we were looking at the thalamus and we were looking at the dorsomedial nucleus of thalamus, we realized that one of the major inputs going into the thalamus was coming from the amygdala. You could see that coming in from the basolateral nucleus. So the basolateral nucleus is getting all of the eventual indirect sensory information. So it's getting information from VPL and VPM, lateral geniculate, medial geniculate, thalamus, then from the sensory cortex, and then that information goes out to the thalamus. So what you can see here is a cortico, limbic, thalamic, and then, as you know, the dorsomedial nucleus of the thalamus goes back up into the frontal cortex. So you have this very big loop among the cortex, the amygdala, and the dorsomedial nucleus of thalamus. Then what we also see is the basolateral nucleus of thalamus is then going to feed into what is called the ventral striatum. Another word for the ventral striatum is the nucleus accumbens, and we're going to be covering this very, very quickly. And what we're going to basically see with the nucleus accumbens is it is going to provide a great deal of information back out up into the frontal cortex. So again, you have yet another loop going on here. But a major output of the basolateral nucleus goes to the central nucleus, and then the central nucleus provides all of these outputs coming. So you can see the central nucleus going to the ventral striatum, going up to the cingulate gyrus, and then going through another huge loop that modulates the hypothalamus. And it modulates the hypothalamus in two major areas. One is the lateral hypothalamus, and then it also modulates the ventromedial hypothalamus. So the amygdala plays all of these very interesting roles. Now, again, the amygdala is very much involved in obviously fear and anxiety and phobias and issues like that. But what it is also involved in and plays an important modulatory role is, again, the ultimate execution of motor responses. And very interestingly, the Graduate Center, where we have our neuroscience program is shared by psychology. That's one of my 10 training areas, called behavioral and cognitive neuroscience. And they, together with biology neuroscience, those two training areas combine to form what is called the CUNY Neuroscience Collaborative, or CNC. A very interesting program that started about 3 years ago, and some of you in the class are members of it, is the Master's Program in Cognitive Neuroscience. So the Master's Program in Cognitive Neuroscience and the CNC hold weekly colloquia. And last Friday, I attended virtually that colloquia. I think there was about 70 people on the Zoom. And basically, we had a person from Mount Sinai named Ivan D. Arojo. And Ivan D. Arojo, his overall interest that I know him professionally over his career obviously has been in ingested behavior. And what he's very interested in is the gut signals, that signals from the gut that then modulate the brain to modulate food intake. And a major area that he is very interested in is the vagus nerve. Now, again, people will just write, and you'll read your textbook, and it'll talk about how the vagal afferents will modulate food intake. Yet, very often, the evidence that you see from direct information coming from the vagus and going up to modulate food intake is not that precise. But using modern neurobiological techniques in which what you're able to do is inject into particular brain areas these adenoviruses. And then these adenoviruses then can get activated within those nuclei by pulses of light, usually blue light. So what De Rojo basically demonstrated, really for the first time of direct influence, is basically looking at vagal inputs coming up from the intestines and the stomach, going through again a peripheral ganglia called the nodose ganglia. We've covered a little bit the sympathetic ganglia. This is a different ganglia, the nodose ganglia, that then will then send fibers further up into the brainstem, where they will interact with the nucleus tractus solitarius and the parabrachial region, and eventually go up into the brain. And what he basically demonstrated was that not only do you have these fibers going up, but then when you activate these fibers, what the animal basically is doing is showing reward types behaviors. So what they basically have in the cage are two little holes, and a mouse is in the cage, and it nose pokes. It puts its nose through one of the holes. When it puts its nose through one of the holes, all of a sudden, a bolt of blue light goes into and activates the nodose ganglion. And what would now happen? This animal would go back and back and back and back and back to get that nodose ganglion stimulated. So one argument that you have here, of course, is that it is reinforcing, positively reinforcing. And further, he then did exquisite studies of basically showing the transsynaptic connections from the nodose ganglion up to dopamine-containing cells. And then basically also demonstrated that in the Nucleus Accumbens, after the animals are doing this nodose ganglion nose poking, there is an increase in accumbal dopamine that is very much associated with the rewarding aspects, not only of that stimulation, but of things like food and of things like drugs of abuse. So he had that side. Now, what he then did was map things down, and eventually what you got was some of these fibers coming back down into the amygdala and into the central nucleus. So now what De Rojo again is interested in is this sort of gut-brain reciprocal interaction. So of course, whenever you have certain kinds of vagal signals, and the vagal signals can be of two types. One is a sort of in the famous Charles Dickens novel, Oliver Twist, where the little orphan boy basically makes the outrageous a request of, sir, I want more. That's appetition. That a vagal signal can basically signal more, or a vagal signal can signal satiety or less. So one of the things that you're basically looking at is, yes, that signal goes in, but what does it basically do? So what now De Rojo then did was work in the central nucleus of the amygdala, and again is doing that adenovirus, where again you get the stimulation with the blue light. And what he basically demonstrates here is stimulus-bound behaviors being driven by the central nucleus of the amygdala. In certain cells in the central nucleus of the amygdala, whenever they get turned on, the animal will stop whatever it's doing, and then all of a sudden take up a stereotypic stance of where they're basically beginning to eat. So they have their little paws holding on to an imaginary food pellet, and the animals are doing rhythmic mandibular and maxillary jaw movements in order to eat. And then as soon as the stimulation goes off, they stop. And this sort of brings me back when I had my dissertation. The third member of my dissertation committee was a very interesting character named Edgar E. Coons, C-O-O-N-S, otherwise known as Ted. And Ted was a music major at Yale, and then was taking some psychology classes, and then was able to get into the laboratory of Neil Miller at Yale. And again, to me, Neil Miller is probably the preeminent physiological psychologist of the 20th century. You can basically look up all of the different areas of work that he did. Now, Neil Miller had such a large lab at the time that people would have to reserve very small amounts of time where they could do their work in the lab. And what Ted Coons was interested in was basically trying to map the hypothalamus for places where if you apply electrical stimulation, the animal will now go into do a reinforced behavior rather than pressing the lever, called intracranial self-stimulation. And his urban legend had it that one Sunday morning at 3 a.m., he was in the lab and he was testing a rat, and all of a sudden, with the parameter of stimulation to an area around the lateral hypothalamus, the animal engaged in stimulus-bound eating behavior. That is, even though the animal is completely full, when this circuit was stimulated, the animal would do all of the stereotypic things of basically eating. And by the way, what he could do is if he stimulated and he put a wood block rather than a food pellet in the cage, the animal wouldn't eat it. It had to be food. And so his great debate was whether to call Miller at 3:30 in the morning and tell him of this, or wait until when Miller would get up at maybe 7 or 8 o'clock. So he decided to call him up at 7 or 8 o'clock, told him the information. Miller then famously asked, well, when did you find that? He goes, at 3:30 in the morning. And Miller said back, why didn't you call me then? Okay. So in some way, what you're basically seeing here is that little quote I gave you a number of lectures ago by Wally Nauda, the question of when does motivation end and motive begin? And what we basically see with these outputs coming out of the amygdala is all types of particular behaviors, aggressive behaviors in which the animal is the aggressor, aggressive behaviors in which the animal is the defender, male sexual behaviors, female sexual behaviors, ingestive behaviors, all start to get driven out of the amygdala, through the hypothalamus, into the midbrain, especially that area of the periaqueductal gray, the dorsal periaqueductal gray, the lateral periaqueductal gray, and then the ventrolateral periaqueductal gray. You can generate a whole set of behaviors that then descend down into the pons and medulla reticular formation that generate reticulospinal types of outputs that engage those basic behaviors. So what we can do is see this here in this sort of drawing. So again, you see all of these very interesting inputs coming from the sensory thalamus, coming from the perirhinal cortex. Remember that having to do with things like smell and whatever, then going to the entorhinal cortex, to the hippocampus, where you have things like one trial fear conditioning. And these things can be driven by what? Aversive unconditioned stimuli, that is a literal shock or something, and then conditioned stimuli, and then also species-specific danger signals. So again, like I'm sitting out here, if right today it's cloudy and back to seasonal temperatures, but if it was an absolutely clear day and I had sheep and donkeys standing outside and the shadow of a hawk comes flying overhead, the sheep and donkeys don't do anything. The chickens all of a sudden stop. They basically have species-specific danger signals. All of this stuff comes through the conditioned stimuli, and then what we basically call, or what Lorenz and von Frisch and people like that would call instinctual behaviors, basically come through the sensory thalamus. And notice with the sensory thalamus, you then have this input going into this perirhinal cortex, entorhinal cortex, and hippocampus, and this information is being generated right into the basolateral nucleus. Now, the aversive unconditioned stimuli also will go through areas like the posterior thalamus, the parabrachial nucleus, and the locus coeruleus. These last two structures ascend back up to the lateral basolateral nucleus. So what is now starting to happen? Unconditioned stimuli that are related to aversion or danger or whatever come in, and then the conditioned stimuli that are either species-specific basically come in, and these two things will now join in the lateral basolateral nucleus. So then the basolateral nucleus then goes to the central nucleus, and look at all of the different areas that get activated. One area is the ventral tegmental area, which of course will then stimulate the mesolimbic and mesocortical dopamine systems. And we'll talk about that a little bit later in this lecture. We also then get the parabrachial nucleus, which is then involved in things like panting, increased respiration, some degree of alertness. Then the central nucleus activates the lateral hypothalamus and eventually goes down into the rostroventrolateral medulla. And of course, that together with the nucleus tractus solitarius projections to the ventrolateral medulla will result in what? Increased heart rate, tachycardia, and increased blood pressure. Then we get into the caudal pontine nucleus of the reticular midbrain, and what you then get is potentiated startle responses. What is now happening? You're priming the sensory systems to a state of hyper alertness. So you're basically looking for small degrees of stimulus change so as to adjust your responses. And then the periaqueductal gray, remember, as I described, the pericanular gray, the periventricular gray, the periaqueductal gray, and then the third ventricle periventricular gray are all parts of the very early nervous system, which basically formulated around the time of the neural tube. And what you can basically see is the lateral part of the PAG will produce non-opioid analgesia and activity bursts. What do I mean by activity bursts? In this kind of case, you're basically talking about what is called agonistic behavior. So I always remember all the way back in the summer of 1971, where I got out of the Air Force after taking a couple of psychology courses out in Nebraska, and I'm about to join this master's program. I decided to take a couple of summer undergraduate courses at City. And one of them was a course called Comparative Psychology, and it was taught by a person who was at the American Museum of Natural History. And so what this woman basically described was, she goes, and now we're going to cover agonistic behavior, AGONISTIC. So then, and she goes, with agonistic behavior is fleeing, fighting, or fleeing. So I sat there, the rest of the class is writing things down, and I raised my hand. I guess simply because I just got through fighting in the Air Force. And then I said, which? And she goes, which what? I go, which one? She goes, it's agonistic behavior, fleeing or fighting. That's what agonistic behavior. And I go, well, which one and how is it chosen? Well, it's agonistic behavior. Of course, literally 15 to 20 years later, I started to read the work of Richard Bandler in Australia, who basically demonstrated that different parts of the dorsolateral PAG, the rostral part of the ventrolateral PAG, was involved in fighting behavior and was primarily glutamatergic. And it's a funny thing, if you're basically studying the animal that you're studying, the rat will do this. And how will they do this? They're right there with another rat. So it's going to be intra-species. So there they are with another rat. In order to begin fighting, the animal who's going to start to begin fighting moves backward. Why? Because what it's doing is putting all of its pressure on its back legs, and they sort of get up like little Tyrannosaurus Rexes and then fight from there, where all of the base of their body is used as a base, and then the animals will fight and bite and whatever. So the actual initial movement in a fighting response is a backward movement to get yourself into an upper position. Conversely, the more caudal part of the dorsolateral PAG, which is primarily GABAergic. So what you're doing now is you have an inhibitor, and what you're basically doing is doing disinhibition, will literally now propel the animal forward, because the animal takes a step forward to basically do a left or right turn to get the hell out of town. They don't want to fight. They want to flee. Okay. But now the other interesting thing, so remember how we talked about this when we talked about the hypothalamus, when we talked about the ventromedial hypothalamus and the work of John Flynn. What basically happened there was that when you stimulated those, or when there were lesions in the ventromedial hypothalamus, you had animals that looked like the Halloween cat. they were always sort of an enraged kind of fighting, spitting, clawing, again, up on their hind legs, because they could use their forelegs to swat at each other, use their teeth and whatever. But the other thing we talked about in the hypothalamus was what? In the lateral hypothalamus, we looked at it from the point of view of the predator, of a cat sort of homing in on a bird or a rodent. And the point is, so now let's look at it from the prey's point of view. That is the ventrolateral hypothalamus is involved in two very interesting effects. One is an opioid form of analgesia, and the second is a freezing response, because in many of the cases, the animal who is the predator is really going to get the prey if they see the prey moving. If all of a sudden the prey stops or whatever, they do this. Now, again, I'm having a senior moment. Have I told any of you about my dog, Hunter? Good. I haven't. I don't think so. But if you heard
No,
it, you'll hear.
you haven't.
Huh?
You haven't. I think you mentioned like a goat or a cat or something.
Well, okay. My dog, Hunter, one night ran out onto our back deck and into our backyard, and he basically came back. She came back with an animal in her mouth, and she dropped it down. So I look at it, and the animal's in the possum. Okay, and I call my sons out there. Of course, I got Hunter away, and I said, "There's an opossum. Look at that." And you saw the opossum lying on its side with its sort of black eye, sort of vacantly staring upward. And you basically saw a little bit of spittle coming out of its mouth. So I take my two sons and I say, "Let's all go inside." We go inside. I go, "Wait 30 seconds. I go, "Okay, come outside." We come outside. We look down and there's no opossum. Okay. And of course, there's the famous saying, for those of you who have never really looked at the thing, there's a saying called playing possum. And playing possum is acting like you're dead. So that and what do you need to do there? You need to literally freeze. You need to stay there very, very still. And then what the predator is done, unless it's unbelievably hungry, may go and do something else. And then go, "Oh, I got that guy. That guy's dead." And then boom, the predator goes away for a second, and the prey goes, "Oh, okay, good time," and they go running off. Now, why else? Look at the other thing that's happening. You have a freezing response, A, to stop your movement, or B, maybe even to play dead. But at the same time, what's being activated? An opioid analgesia. Remember all of those lateral spinothalamic tract fibers coming up and going, pain, pain, pain. They got to the ventrolateral PAG, and the opioid synapses in there basically send descending signals that inhibit the pain. Because what you don't want to do is go, the opossum sit there, "Well, I just got dragged by this shepherd-like dog up here. I know my back of my neck is hurting me. Oh, my toe must be damaged. Let me clean my toe." Well, you do that, the predator is on you again and kills you. So what you've done is had opioid analgesia. And probably one of the most interesting things that I always think in the history of sports was the famous, and I think the greatest heavyweight boxer of all time, Cassius Clay, who then became Muhammad Ali, fought, and he was in a fight in the early 70s against a guy named Ken Norton. And Ken Norton, in the first round of the fight, broke Ali's jaw. Ali fought for 14 more rounds. And the point is that what you got there is what is called stress-induced analgesia, one of the things I first studied that sometimes can be blocked by opiate antagonists, but not. But what you can see coming out of the amygdala is this unbelievable array of all types of parasympathetic responses, motor responses, and then enhancements of other sensory responses, and then the elicitation of complex behaviors, basically coming out of the amygdala. And notice that, yes, some of this information is coming in, but it's not coming from the prefrontal cortex telling the amygdala to do this. Some of this is being done before some of the information has even gotten up to what we call the prefrontal cortex and the decision-making area. So there's an enormous amount of importance as to how this area of the brain works. And then when we start to think about dysfunction like PTSD or various types of impulsive behaviors that can be attributed to the amygdala, what we can then basically see is that importance. So again, here's yet another type of drawing of looking at the behavior and how the amygdala, the basolateral and central nuclei, both modulate other ventral tegmental input or modulate this nucleus accumbens and ventral striatum. Okay, another very major structure in the limbic system is called the septum. And where is the septum? Well, one thing you basically learned all the way back in gross anatomy is when we were looking at the anterior horn of the lateral ventricle, or what we actually were doing was looking at what? The anterior horns of the lateral ventricle, left and right. They were separated by a thin sheath of cells called the septum pellucidum. So now, as we move caudally, and we're going to see this when we look at the brain in coronal sections, as we move caudally, what starts to happen is the lateral ventricles start to move. They stop that horn-shaped look and now start to form the body. But right around there, the septum pellucidum gets thicker, and we have the septal nuclei. And there's the lateral septal nuclei and the medial septal nuclei. And it's a tiny little sheaf of cells. Yet, what I'm basically telling you to look at is look at all of the inputs, all of these areas, the hippocampus, the medial septum, the VTA, the raphe, the locus coeruleus, the lateral dorsal tegmentum, the PAG, the thalamus, the bed nucleus of the stria terminalis, the amygdala, the prefrontal cortex, and the entorhinal cortex all provide input into the lateral septum along with the hippocampus. And then the lateral septum projects reciprocally to all of these areas and projects to the Nucleus Accumbens, by a pure, just as a pure output. So this area was basically studied by Walter Hess in his Nobel Prize winning work, in which he basically placed lesions of the septum and basically found that these animals, cats that he was studying, engaged in what was called septal rage. They basically were hypersensitive to light, hypersensitive to touch, hypersensitive to sound. And they would, as a result of that, would fling themselves into a unbelievable kind of aggressive types of behaviors, even though nothing was there. Further, this lateral and medial septal areas then, of course, had reciprocal connections with the hypothalamus and had reciprocal connections with the amygdala. So in the late 1930s, it was thought that the septum was engaged in rage behavior. We found the area of the brain, the septum normally was there to calm people. And that loss of the septum, with a septal lesion, would now produce what? This rage behavior. In also the late 30s, two Swiss researchers named Kluver and Busey did bilateral lesions of the amygdala. And they did this work in rhesus monkeys. And so basically, when you had rhesus monkeys in captivity and in individual cages, basically the person who was taking care of them would walk through, and all of a sudden, what would happen is a rhesus monkey that's sitting in the back of the cage comes running forward, grabs its arms and its legs, and then is shaking the cage, screaming, right? And having all kinds of aggressive types of behaviors. Kluver and Busey did these amygdala lesions, again, late 30s. And all of a sudden, at the end, after that lesion, what they could basically do with the monkey is literally open the cage, reach out, the monkey would take its hand, come down, and they would walk down a walkway. You wouldn't have to put the monkey into a monkey chair and whatever. And so the argument is that the amygdala, the basic thing of the amygdala was aggressive behavior, and that lesions placed in the amygdala produced calm. So again, this whole thing of where I began today's lecture with the idea of certainty, with the idea when we looked at the medial and lateral hypothalamus, and we looked at a differentiation of predatory versus intraspecific aggression, when we looked at, quote, hypothalamic feeding centers, the same sort of thing was thought of with the limbic system of the septum and the amygdala. The interesting thing is, for a while, these folks basically believed this, but one of the things that was very interesting about these monkeys is, so here is an animal with a amygdala lesion, and they are very calm and whatever, but what would they end up doing? They'd be ending up being hypersexual, to the point where, as the researcher would walk down a runway with the monkey, all of a sudden the monkey would grab the researcher's leg and start humping the leg on the thing. So, this idea of a simplistic center for a complex behavior, held sway until it no longer did. And this becomes very important. Yes, the septum and the amygdala play a role in the modulation of these things, but the simple destruction of one versus another is not going to eliminate what you would think to be an unwanted behavior. So now we focus on the Nucleus Accumbens. So the full name of the Nucleus Accumbens is the Nucleus Accumbens septi. So what basically you're looking at is the Nucleus Accumbens, which you can see here in yellow, is here and here. Here's where the septal nuclei are. And just its very simple name, the Nucleus Accumbens septi, makes it an argument that it is part of the septal nuclei and hence a part of the limbic system, an artificial contrivance. Also, look at this. What is right here? Here is the caudate. Here is the putamen. And here is the globus pallidus. All three of these structures are the basal ganglia. And very often, we usually use the word dorsal striatum. So the nucleus accumbens was also known as the ventral striatum. So the question then became, is it part of this artificial delineation called the limbic system, or is it part of that artificial delineation that we call the basal ganglia? And the whole point is that it turns out that it's both. And that basically what you have in the ventral striatum, in the nucleus accumbens, is yes, it has some very important motor components, because as we're going to see, it's going to project out into the ventral pallidal nuclei, and eventually there's going to be motor output. But it's also incredibly important in motivational input. And that what now becomes important is that there are two parts of the nucleus accumbens. The nucleus accumbens core, which is the center nucleus, and then on its medial edge is a sort of boomerang shaped structure medially called the nucleus accumbens shell. So here is that question is, if you look at this diagram, what you can see is here's the caudate and the putamen and globus pallidus, and here's the nucleus accumbens, and we can call that the striatum. And the globus pallidus, of course, is then going to send motor output, and the nucleus accumbens, a major output to it, goes to the ventral pallidum, which goes to motor output. So is this a motor or a limbic structure? And again, we have the question, where does motivation end and motor begin? And then the whole idea of this circuit of dorsal striatal circuits, ventral striatal circuits, limbic circuits comes to that great neuroanatomist Meatloaf, who sang all revved up with no place to go. Okay, so here we're looking at the nucleus accumbens. And again, look at the types of, we're looking at three major neurochemical outputs. I should point out that that sort of minimizes a fourth major neurochemical input or output called acetylcholine. Why? Because 80% of nucleus accumbens cells that are found within the nucleus accumbens are called medium spiny projection neurons. And over 50% of them contain the neurotransmitter acetylcholine. And so what you're going to have is excitatory output, output coming out of the nucleus accumbens up to the frontal cortex. It also, the nucleus accumbens will also innervate the basolateral amygdala. And they do that primarily through a cholinergic output, which is 99% of the time excitatory. However, the nucleus accumbens other major output, with the remaining number of output cells that are not interneurons, is a major GABAergic projection to the ventral pallidum. And the ventral pallidum is very much involved in behavioral motor output, where you basically see the animal now engaging in a behavioral response. Notice what the ventral pallidum does to behavioral output. It is exerting a GABAergic influence, which means what? That the ventral pallidum, under normal circumstances, will inhibit behavioral output. The Nucleus Accumbens, because it's connected to the ventral pallidum through a GABAergic input, inhibits the ventral pallidum. So what is it doing? It is disinhibiting behavioral output. So now, when the Nucleus Accumbens is active, it is producing an inhibitory effect on an inhibitor of behavioral output, thereby allowing a gate through which the behavioral output goes. So now the animal goes and behaves. Now, what I want you to basically think of here is two particular drugs. What I want you to think about is the drug called amphetamine and a drug called cocaine. So both of them work, albeit in different mechanisms, upon dopamine function. In the case of cocaine, it acts as a psychomimetic, and it also acts to release normal amounts of dopamine. So now, with amphetamine, in an amphetamine-treated rat, you're going to have more dopamine being released, and there is going to be amphetamine working on dopamine receptors in the accumbens. In the case of cocaine, cocaine works through dopamine transport by inhibiting dopamine transport. So now what do we know is that when dopamine, as a typical classical neurotransmitter, is subject to what we call reuptake, that about 95% of the dopamine that gets released from a BTA neuron will eventually get reuptaken through dopamine transporters. But if you give the animal cocaine, the cocaine inhibits the dopamine transport reuptake mechanism. So what does that do? It puts more dopamine in the synapse for much longer periods of time. So now here's a guy on amphetamine or cocaine, and the nucleus accumbens is getting stimulated with dopamine. It then gets activated through a disinhibitory thing that inhibits the ventral pallidum, that releases behavioral output. So what do you got with a person with a person with cocaine or amphetamine? They're highly stimulated. They're doing lots and lots of motor acts. They may be doing lots and lots of impulsive motor acts. They're doing the motor acts when they shouldn't be doing the motor acts. So they're doing all of those kinds of behaviors. In the case of a rat, you have a rat running all over the goddamn place. So what you have there is that effect. Now, in addition, from the BTA, you can see that there is two different kinds of pedals. That is there's a dopamine pedal and it's a GABAergic pedal. Remember, 50% of the BTA neurons are non-dopaminergic. So that inhibits and inhibits this. So that can't fire as much. If that can't fire as much, this will fire, and therefore you'll inhibit behavioral output. So there's this step back and forth. But if you're now giving amphetamine or cocaine, this system is not getting activated. This system is getting overactivated. Also, the mesocortical projection to the frontal cortex is getting overactivated. So therefore, the frontal cortex comes back down through a glutamatergic input into the accumbens. The hippocampus has a glutamatergic input. The thalamus has a glutamatergic input. The thalamus stimulating the frontal cortex that stimulates the accumbens has a further excitatory input. And the BLA, any kinds of information that is anywhere salient, all of a sudden gets to be acted on. So what you can see very importantly is the importance of the nucleus accumbens in the ventral striatum in modulating not only behavioral output, but getting all of the relevant information from cortical, limbic, thalamic, and then brainstem structures. So now the other interesting aspect about this is the fact that the nucleus accumbens is a structure that when you put cannulae, these little tubes, into the nucleus accumbens, and you place into the tube through the cannula drugs of abuse, that virtually every major drug of abuse, if placed into the nucleus accumbens, the animals will avidly self-administer the drug into this site. So that goes for opiates, that goes for cocaine, that goes for amphetamine, that goes for nicotine, that goes for caffeine. And the other interesting thing is that when you do this, and then you do a rank ordering of the potency of the particular drug to produce this drug of abuse, the amount of dopamine that gets released is different. So for opiates, or for cocaine, or for amphetamine, you may get a 200 to 300, 2 to 3 time fold increase of extracellular dopamine in the nucleus accumbens after that microinjection. Then when you move down to something like nicotine or caffeine, there might be an increase of 50 to 100%. Then if you move down to the active ingredient of that wonderful thing, we've had Halloween. So here you go. That wonderful thing called chocolate. You can get a 50% increase in dopamine if you put the active ingredient of chocolate into the nucleus accumbens. So what we basically see is this reward circuit of the VTA and nucleus accumbens. But now when we look at it, we have to basically ask, when in the world are we, whatever drug that we're getting, where are we in terms of drug reinforcement or drug dependence? So what you can see with initial taking of a drug, and this is the work of Nora Volkow, who was the head of NIMH, and George Koob, who is now the head of NIDA. And what you basically see with initial administration or opportunity for the drug to self-administer, reward circuits like the VTA and the nucleus accumbens are activated. And that what you then get is very drug reinforced memories, because you can do this by administering the drug in one half of a cage that has, let's say, vertical stripes and a flat floor. And then you inject saline into the other part of the cage, which has a sandpaper floor and horizontal stripes, and then vice versa. And then where the animal got the drug goes into the memory in the hippocampus, and the animal will do what is called a conditioned place preference and stay there. Okay, so you're engaging the hippocampus. you're also engaging the conditioned responses under which the animal got the thing. So the animal will actually do superstitious behaviors, going, boy, all of a sudden it's not like, hey, you just got cocaine into your accumbens. No, the animal will now then seek out and do these conditioned responses again. So in the case of pressing the lever and getting it, it will press that lever again, or stay in that part of the cage, or do particular stereotype behaviors that have nothing to do with it. But then over time, when you start to have this reward circuit, you also then create what is called craving, which is the expectation of a drug. Well, I got it now, and by the way, this guy comes along, picks me up, puts something on my head, I go into a cage, and the last 3 days, whenever I pressed this lever, when a light came on, I got food. So now, even in the home cage, waiting to be put into the test cage, the animal is now using, exhibiting craving and expectation of the drug, which is increased activity in the cingulate, prefrontal, and orbitofrontal cortex. But then over time, what now starts to happen is, if given the opportunity, an animal can binge and have a temporary loss of control. Oh, I'm not going to do it. I'm not going to do it. I'm not. How many movies have you seen where you see it and then finally, boom, something bad happens and poof, the person goes into a binge, where again, you have reward circuits, the VTA and the Nucleus Accumbens happening. And what happens to top-down control? The frontal cortex, the descends, decreases. What do I mean here? Again, that movie that I suggested that you all go and see at some point in your life with Woody Allen, called Everything You Wanted to Know About Sex. What you basically have is the situation in which there's a Woody Allen character sitting there, and you have, the little angel over here, always usually on the right side, and then the little devil on the left side, talking. And in here, you're basically saying, well, if there's top-down control and you don't want, you want control, you want ego-related, reality-related types of things, you should have the frontal cortex saying, do this, do this. And what you're doing is you're trying to block the drug expectation here. Damn it. You're trying to block the drug expectation here. So you're trying to control, and then boom, the little devil gets through, and boom, your VTA, your accumbens, your amygdala goes, go do it, and boom, you binge. But now over time, with this repeated situation, you're now moving to another place called withdrawal. And in withdrawal, you now don't have the drug. And by the way, what has now happened to the reward circuits because of repeated taking of the drug? There is now a change in here. So one of the things you want to do is you want to cycle, because a number of these drugs have tolerance effects. So if they have tolerance effects, what do you need? You need more of the drug, which means you need to binge more than just take the normal amount, or you suffer withdrawal effects. And of course, over time, the rewarding aspects of the drug from chronic use versus initial use at a particular dose goes down, along with the anterior cingulate and the prefrontal cortex get activated. So you now cycle right past that drug and back to craving. Withdrawal increases craving, craving increases binging, binging increases withdrawal, that increase, and it's a vicious cycle. But it's no longer a cycle that's driven by reward. It's driven by what? Avoidance of the untoward effects of dependence. And we see this very, very powerfully in a whole bunch of different neurotransmitters during different parts of drug things within the accumbens. So here's a nice little map of looking at, look at that huge list of the Nucleus Accumbens shell. The Nucleus Accumbens core does it, but the shell seems to be very much affected by much more the initial reinforcing effects. Also, the central nucleus of the amygdala, some for the bed nucleus of the stria terminalis. So from the shell all the way through the bed nucleus of the stria terminalis out to the central nucleus of the amygdala, we have this big long swath of cells that seem very, very affected by the drugs of abuse.
Wait, Professor,
Yes.
quick question for all the addictions and stuff on the last slide. Ethanol, that's alcohol, right? Like alcohol.
Yes, that's alcohol.
Okay.
Yes. Yes, I'm sorry. Yep. Anybody, and THC, of course, is marijuana, the active ingredient of marijuana.
Okay. Thank you.
Yeah. And then the list goes on and on and on with a whole bunches of drugs of abuse. Okay.
Yeah. Thank you.
By the way, very good for interrupting me. Other people, let's see, you can be asking me as well. Okay. So now we switch to another structure in the brain, which of course is called the hippocampus. And look at the name hippocampus. Basically, it was described by Latin and Roman neuroanatomists, and basically were influenced by the sea creature that would be swimming around in the Mediterranean called the little seahorse. So it had this very interesting kind of shape. And the hippocampus has this interesting shape. So you have a dorsal region, which is called the CA1 region. You have a curved region, which is called the CA2 region. We have a ventral region, which is called the CA3 region. And then what you then have is the CA3 region goes into this sort of mouth-like structure called the dentate nucleus of the hippocampus. Some people call that CA4. Now, what the hell, if I'm talking about the hippocampus and I am talking about these regions of the hippocampus, why do I use the word with the literal name CA? The reason is that when you look at the two hippocampi next to each other, they're mirror images of one another, and what they literally look like are goat horns or cow horns or any kind of horn coming out. And of course, it looked like a horn to the Roman neuroanatomist who gave them the name. The name of that Roman neuroanatomist was Ammon, A-M-M-O-N. So therefore, Ammon didn't say, hey, these are Ammon's horns, because that's English. What did he say? These are cornu, C-O-R-N-U, which means horn, ammonis, cornu ammonis region 1, cornu ammonis region 2, cornu ammonis region 3, and then the dentate nucleus. So that's where those terms come from. Now, all the way up till now, when we have basically talked to you about very interesting nuclei that modulate behavior. So we talked about the locus coeruleus, and I talked about that these cells are all of the cytoarchitecturally described cells of the locus coeruleus, all synthesized and release norepinephrine. But it was a nucleus, and then different parts of the nucleus would project 6 different directions. And I described that. Then I described the raphe nuclei. Then I described the nuclei of the substantia nigra pars compacta that form the nigrostriatal tract. Then we described the dopamine nuclei, 50% of them are the ventral tegmental area that would either do the mesolimbic projection or mesocortical projection. Then we described all of the amygdaloid nuclei and how they were interconnected with each other, and the differential amounts of inputs and outputs that then defined how that amygdala would work. In all of these cases, what did we have? We had what you would learn in a Psych 101 lecture about a thing called the neuron. That is, the neuron would have a soma, and coming off of the soma would be these little areas called dendrites, and that the dendrites and the soma had receptors on them. And this was the input coming into the neuron. Then at the axon hillock, there was a temporal or spatial summation of the activation or inhibition of those receptors being occupied, so that we move from a resting state to an action potential. And then at the end of the neuron, we then have the axon, we have the release. So basically, you have input, you have synthesis within the cell, and then you have an output, right? And that goes on for the thousands or tens of thousands or hundreds of thousands of cells of all of the nuclei we talked about. But now we come to something completely different. And again, this is a phylogenetic type of advance. What we're going to be studying simply in this course is 3 structures: the hippocampus, the cerebral cortex, and the cerebellum, that do not, all of the cells within them do not conform to the input, synthesis, and output of all these other cells. Rather, you have a stratification of function so that you create hippocampal layers. Some of the layers are there to do what? To gather in the inputs. Then what we have is some of the layers are there to synthesize the response. And then finally, there are layers there to send out an output. So in the hippocampus, not only do we have 4 regions of the hippocampus: CA1 dorsal, CA2 curve, CA3 ventral, and then within the hippocampus, the dentate gyrus. But throughout all of these regions, we have 6 layers. The outermost layer is called the molecular layer. So now don't get confused. The outermost layer, layers 1 and 2 of the hippocampus, are going to be on the dorsal surface at CA1. They're going to be on the medial surface of CA2. They're going to be on the ventral surface of CA3. And then they're going to be on the dorsal and ventral surface of the dentate nucleus. So those are the molecular layers, and they are, and layers 1 and 2 are called the subiculum. The molecular layer are cells. Layer 2 is striatum lacunosum. What is that? Those are fibers that are entering into the hippocampus. Okay, so the layer 2 are the fiber inputs that are coming into layer 1 and also affecting layer 3, the striatum radiatum, which is the dendrites of the pyramidal cells. And the pyramidal cells of the hippocampus are what? They are the output cells. So what types of pyramidal cells are they? They typically have an apical dendrite. That is a dendrite that shoots up and then has bushes out. So the inputs are all coming in there. So you're either having inputs onto the dendrites of the pyramidal cells, or there's going to be inputs in the striatum radiatum, which is going to touch the pyramidal cells themselves. And then through a spatial or temporal summation, the pyramidal cell is going to fire. Layer 5 is the stratum oriens, which are a group of molecular types of cells in layer 5. And then the alveus is both cells that are coming in inversely, or they are going to be axons of the pyramidal fibers that are now exiting the hippocampus. So there is a stratification and a separation when we get to hippocampal layers, and then when we see it again with cortical layers, and then when we see it again with cerebellar layers, of a differentiation of function between inputs, between integration of the inputs, and then finally outputs. So what are the major afferents coming into the hippocampus? So again, an afferent is an input. And the major hippocampal afferent is called the perforant path, PERFORANT. And this is a major kind of fiber input coming in from the entorhinal cortex. So again, let's go back to Mortimer Mishkin, who basically talked about dorsal stream inputs and ventral stream inputs as they have to do with memory, where he basically talked about dorsal stream inputs having very much to do with spatial memory, and ventral stream inputs having to do with episodic or semantic or event-related memories. So all of this entorhinal cortex is giving all of these tertiary visual, tertiary auditory, tertiary somatosensory inputs coming into the entorhinal cortex, and the entorhinal cortex feeding into the hippocampus. Now, like I said, the typical way you do it is information comes in through the subicular layer and then filters down to the pyramidal cells, and then you have the alveus coming out. But there is inputs coming in from the other end, coming into the alveus, and that's coming. And what is the fiber tract that is eventually formed by the hippocampal outputs? It's the fornix. But there are inputs coming into the fornix that is called septo-hippocampal inputs. So these are inputs which originate in the septal nuclei and then go back to the hippocampus. They're going, the fornix, you think, is always going caudal to rostral, then it makes a turn, and then it goes back to the mammillary bodies. Here, you have, as you move forward and you're about to make the turn, that's where the septum is. The septal nuclei send inputs back into the fornix. Now, what did I tell you about the Nucleus Accumbens? What I told you was that you had what are these called these medium spiny neurons, and about 50% of them are cholinergic. They produce excitatory effects. And again, the septum, as we can see, is involved in lots of different types of social behavior, because remember with Hess, we destroy that and we have a cat who's nuts, right? Just reacting inappropriately to simple stimuli. So the septo-hippocampal pathway, which is primarily cholinergic, gives cholinergic input into the hippocampus. Now, one of the things that becomes very clear in the etiology of Alzheimer's disease is agitation and confusion, where all of a sudden a person who is suffering early Alzheimer's symptoms all of a sudden becomes disoriented. Oh, I'm here, I'm thinking I'm here. My late father-in-law, in the last year of his life, had definite Alzheimer type symptoms. So there was one day he lived out in central New Jersey in a town called Chatham. And to the west of him was a town called Morristown. And what he would do is he'd go to Morristown, go to a library to look up things and stuff like that. So one day he goes to Morristown. He gets into his car and starts driving. Except which way was he going? He was going west instead of east. And he drove for like 100 miles, and thankfully the car ran out of gas and wasn't sure where he was. Eventually people called, checked home, and realized he went the wrong way. Of course, what was his reaction to the whole incident? It wasn't like, oh, I was confused. The car is terrible. It ran out of gas. It made me go the wrong way. So therefore he sold the car. Okay. So I mean, that's a distillation of something very, very interesting. And what eventually was found through the monumental work of one of the acolytes of Solomon Snyder, if you basically go and you look at all of the major neurotransmitters, and you look at people, especially figuring out the receptors of those neurotransmitters, and there's multiple receptors for each of the neurotransmitters, whoever made that discovery was either Snyder himself, or it was one of Snyder's graduate students who went on. I think Solomon Snyder had somewhere between 6, I'd have to sit down and count them one day. I think he had 6 or 8 of his students that eventually became presidents of the Society for Neuroscience, an incredibly influential person. But one of the people that worked with him was a guy by the name of Joseph Coyle. And in the early 1980s, Coyle, based upon these studies, basically talked about the cholinergic inputs coming in from the septum into the hippocampus, becoming depleted, and that this was an early marker of Alzheimer's disease. And that what you could basically do is look at an animal model showing these things, in which the amount of cholinergic depletion in there was an incredible predictor of the ability of the animal to basically go through this. So, so here we have these medium spinal projecting neurons, cholinergic neurons of the septum. And then, as we talked about, the nucleus accumbens septi, those outputs that are controlling those ventropallidal outputs and controlling the frontal cortex and whatever, mediating the effect, and that there's a cholinergic effect here. Then what we also have, in addition to the entorhinal cortex, is an enormous amount of other inputs coming into the hippocampus, that is called together a Greek sounding name, the psalterium. Now, in addition to that, we then have major hippocampal inputs coming from the locus coeruleus, providing noradrenergic inputs, and as I mentioned, the control of hippocampal theta, which is a very prominent type of EEG response that you see during sleep, during what is considered to be memory consolidation. And then serotonergic types of inputs that are coming into the hippocampus. And then the major efferent of the hippocampus is the fimbria, which we're going to see when we're looking. As you see, you have, remember, the alveus is what? Covering what? The dorsal surface, the curve, the ventral surface, and the whole dentate nucleus. So all of these outputs, as they're coming out of here, is all splayed. So that's a fan-like shape, and that's the fimbria. And then eventually the fimbria tightens up and forms that fiber tract that we all saw in gross neuroanatomy called the fornix. So what you can see here with the hippocampus is you can see that there are connections, reciprocal connections with the entorhinal cortex. You can see the fornix. The fornix also goes, innervates the prefrontal cortex. The anterior thalamus and midline nuclei innervate the prefrontal cortex. The prefrontal cortex has reciprocal connections with entorhinal cortex that then feeds into the hippocampus. The prefrontal cortex has reciprocal connections, with perirhinal cortex, smell brain, the other association areas, and then the parahippocampal cortex has connections. And then you have the other inputs from retrosplenial and cingulate cortices, all coming into the hippocampus. So now a number of different things were basically done in order to go and study the hippocampus and memory. And one thing that basically would be studied is basically looking at the perforant path, which is associative long-term potentiation. And the perforant path, of course, is a pathway that's going into the hippocampus. And then you have a Morse fiber area from the dentate area to CA3, which is a Morse fiber, which is non-associative LTP. And then what you have is interactions between CA1 and CA2 and CA3. So now what you're going to be doing is you're going to be recording from CA1, and you'll either stimulate the perforant path or you'll stimulate the Schaefer collateral pathway with trains of stimulation. So basically, here all of a sudden is a burst of stimulation. The very first time you have a burst of the stimulation within a group of cells, and now you're recording, you of course get this powerful, powerful spike of EPSPs, excitatory postsynaptic potentials. And then what you can see is that activity, because it's such a huge suprathreshold spike, not only has a peak effect, but what basically happens, what you basically have is some activity that lasts for a fairly long period of time. Look at that, 2 hours. You can see this activity going on. So you've really changed the way the hippocampus is responding. But now what can you do is you can do this stimulation not one time, but you now do this stimulation, 4 times. And now all of a sudden by the 4th time, notice that the peak is now higher. It's also later, but all of a sudden the activity has now changed. And what LTP is long-term potentiation. That basically the CA1, CA2, CA3 dentate group of cells start to fire, and the LTP is an artificial substitute for what? It's an artificial substitute for external stimulation coming in through the perforant path or any kind of other input into the hippocampus. And so what you're now doing is getting synaptic strength and synaptic change. So that in this case, by going from 1 to 4 trains, you're now doing this temporally. You can also do this spatially. So now the whole characteristic of these hippocampal cells that get selectively activated are now changed. Their synaptic strength has increased, and all of a sudden, so now what do you have? You now have what you call a short-term memory, okay? You've literally taken stimuli and now translated that into memory, and that memory then persists over a bit of time. Now, that is the LTP, and the person, of course, who studied that was O'Keefe, and basically won the Nobel Prize for this, for a neurophysiological correlate of hippocampal memory. Now, the second area that basically can be studied is, again, this arrangement of CA1 dorsal, CA2 curve, CA3 ventral, and dentate nuclei serve another purpose. All of these things are interconnected with one another. But what you could basically find in using a paradigm originated by a gentleman named Morris. So whenever you ask who's buried in Grant's tomb, it's Grant. Who created the Morris water maze? Morris. Morris basically took rodents and he put them into a tank of sort of milky colored water. So you couldn't see the bottom, you couldn't see things, and the tank had homogeneous sides to it. So the mouse or the rat would be put in the water, and the mouse would swim around and swim around. Now, in the water, slightly submerged, was an upside down little flower pot. And the mouse would eventually find it. And he would get onto the flower pot. Now the animal didn't have to swim or tread water anymore. It could stand. And of course, if you kept putting the mouse into the same place in the water maze, what would happen is the animal would do a more and more direct route to the flower pot. Then you put the mouse in different parts of the water maze, and the animal would eventually learn where the flower pot was, okay? And what you could basically do is correlate that with hippocampal activity. And what you basically found was that there were cells in the hippocampus, ensembles of cells in either CA1, CA2, CA3, or dentate. So you would have like triads of cells. So there'd be a cell in the dentate, a cell in CA3, and a cell in CA1. And they would increase their activity as a result of a successful finding of the of where the flower pot was. And they ended up being called hippocampal place cells. Now you can see there are hundreds of thousands of hippocampal cells. And what you could do is use the same cell over and over again, pairing them systematically with other cells in CA3 and the dentate to create hundreds of millions of little maps. So now what we have, in addition to when we talk about the hippocampus in all other kinds of memory, we talk about it as an important structure in memory of place, memory of space, memory of procedure, and spatial memory also works here. And of course, in each of these cases, a number of these things are modulated by cholinergic mechanisms, and they're also modulated by noradrenergic mechanisms. Okay? The last thing that I want to point out with the hippocampus is the hippocampus also has a very, very rich store of steroid receptors. So they have mineral corticoid receptors and glucocorticoid receptors. And again, it makes the hippocampus then a very susceptible and very responsive to stress-related things. So all of a sudden you want to put your memory into hyperdrive if you're stressed. But then if you're overstressed with chronic stress and everything, you then can see a role for the hippocampus in things like PTSD, etc. So that is my little quick review of the limbic nuclei. So let's see what we have in the chat here for a second. This is great. Thanks, John. Okay. Oh, I see that more has to do with the paper. Okay, I see. Very good. So does anybody have any further questions about the limbic system? So at this point, let me see what time is here. Good. Let's take about a 5-minute break, and then I will move into the basal ganglia. So let's just take a little break here. Okay, so I'm back. And so now we will now go into the basal ganglia. Okay, so we are now going to be talking about the basal ganglia or the dorsal striatum. So when we're doing this, we're paying attention to three major nucleus groups. One, which is called the caudate nucleus. The second, which is called the putamen. And then the third, which is called the globus pallidus. So, and the globus pallidus is divided into a lateral or medial or external or internal set of nuclei. So the basal ganglia is made up of the caudate, the putamen, and the globus pallidus. The caudate and the putamen are collectively known as the neostriatum. So therefore, neo, which means newer, and then, so therefore, by default, the globus pallidus is known as paleostriatum, older. Okay. And when we basically look at this, we see that the basal ganglia has a lot of connections with the substantia nigra. It's going to have connections with the thalamus. It's going to have connections with the cortex. And then also interactions with deep cerebellar nuclei. And all of these things seem to have an important thing having to do with movement. So here, what we can see, where we look at the caudate nucleus, what we learned about was that what we have are the lateral ventricles. And the lateral ventricles can be then divided into the anterior horns, which is primarily in the frontal lobe, the body, which is primarily in the parietal lobe, and then we have the occipital, we have the posterior horn of the going to the occipital lobe, and then we have the inferior horn of the lateral ventricles that go into the temporal lobe. And then if we basically look at the caudate nucleus, we basically have a tadpole type of shape to it, so that the sort of head, the thickest and largest part of the caudate nucleus, can be found on the lateral surface of the anterior horn of the lateral ventricle. Then we can see that the body of the caudate nucleus can be found corresponding to the lateral surface of the body of the lateral ventricles in the parietal lobe. And then we can see the tail of the caudate nucleus forming much, much thinner, forming the lateral surface of the inferior horn of the lateral ventricle. So again, I always say the caudate and lateral ventricle relationship. Mary had a little lamb, its fleece was white as snow, and everywhere that Mary went, the lamb was sure to go, with the lateral ventricle being Mary and the lamb being the caudate nucleus. So now, when we're looking at the neostriatum, we see the caudate nucleus, and we see the putamen. That one very important thing that we see here is that this large set of cells in the midbrain, the substantia nigra pars compacta, of which 50% of the cells are dopaminergic, and that the vast majority of those dopaminergic cells of the substantia nigra will form the nigrostriatal tract. And the nigrostriatal tract projects forward up into the caudate nucleus and into the putamen. So the dopamine input going into the basal ganglia is going into the caudate nucleus and the putamen, or going into the neostriatum. So now, the other important thing to think about when we look at this, and now what are we doing? We're looking at a cross section, or a transverse section of the brain. And what we can see right here is we can see the lateral ventricles right here. And then what we're beginning to see here is the beginning of a third ventricle here. Okay. But what we basically see is the caudate hugging the what? Lateral surface of the lateral ventricle. And the caudate and the putamen are these dorsal striatal structures called the neostriatum. And they are called the neostriatum because the cytoarchitecture of the cells in the caudate and the cytoarchitecture of the cells in the putamen are literally identical to one another. So we put the caudate and the putamen together to call it the neostriatum, based on cytoarchitecture. You can also say the caudate and putamen are placed together because they both receive this nigrostriatal dopamine input. Then what we have is the third component of the basal ganglia, which is called the globus pallidus. And the globus pallidus, because it has different types of cells cytoarchitecturally, is called the paleostriatum, simply because it existed earlier in very ancient, in lower animals, where you basically don't see a neostriatum. And the globus pallidus is divided into a pars externa and a pars interna. And what separates them is the medullary laminae of the globus pallidus. So there's one medullary lamina that separates the putamen from the globus pallidus zona externa, and there's a second medullary lamina in the globus pallidus that separates the zona externa from the zona interna. So now I'm going to come up with 7 terms. We have the basal ganglia, which causes the caudate, putamen, and globus pallidus. We then have the caudate, the putamen, and the globus pallidus. Okay. So now we are up to 4 names: basal ganglia, caudate, putamen, globus pallidus. Now a 5th term is neostriatum, which is the caudate and putamen put together based on cytoarchitecture. A sixth term is paleostriatum, because it separates the globus pallidus from putamen and caudate. And then finally, we have a seventh term, which is called the lenticular nuclei, LENTICULAR, the lenticular nuclei. And what are we doing there? What we're pairing is the putamen and the globus pallidus, and we're separating them from the caudate based on location. So whereas the caudate can be found on the medial edge of the internal capsule, the lenticular nuclei, otherwise known as the putamen and globus pallidus, are found on the lateral edge of the internal capsule. And of course, what is the internal capsule up here where we're looking? We're looking at the anterior limb of the internal capsule. So what are these fibers that are coursing through the anterior limb of the internal capsule at this point? These are corticospinal fibers. So it's sort of like a big boulevard where you're exiting the cortex, whereas when we get to this is the anterior limb of the internal capsule, the posterior limb would be having somatosensory fibers. So we have these seven terms that subdivide all different aspects of the globus of the basal ganglia based on cytoarchitecture, based on nuclei, and based on location. Okay, so now the next important thing to think about here is to examine the basal ganglia in relation to two other major structures, one that we have already studied and a second that we'll study a little bit later. And that is the thalamus and the cortex. So depending on your point of view, and if you notice this, you have arrows. The arrows go from the cortex to the caudate and putamen, the caudate and putamen to the zona externa and zona interna of the median eminence, then the zona interna of the not median, the zona externa and zona interna of the globus pallidus, then the zona interna of the globus pallidus projects to the thalamus, and then the thalamus projects to the cortex. So we can refer to this, depending on whatever vantage point you're looking at from, as a cortico-striato-thalamic circuit, a striato-thalamo-cortical circuit, or a thalamo-cortico-striatal circuit. Now, a lot of people may think about it from a thalamo-cortico-striatal circuit, because what are you looking at there? You're looking at that from a sensory standpoint, right? Because all of the sensory information, that is visual information, auditory information, somatosensory information, enters the thalamus, then goes up to the cortex, then you come back out through the basal ganglia, and then the basal ganglia may influence another group of thalamic nuclei that eventually will end up with motor movement, and the movement will come either from the cortex corticospinally, or it will come out of the globus pallidus zona interna through the subthalamic nuclei, which is called extrapyramidally. So in addition, what we also have, in addition to this, and of course, a cortico-striato-thalamic circuit is an argument, which is a vitalistic argument, that the cortex in the famous words of George W. Bush, is the decider. I'm going to decide when we move, and this is what we're going to go and do. And then, of course, from a striato thalamocortical thing, you're looking at extrapyramidal control going on here. So then when you're looking at that, you're looking at dysfunction, the major dysfunctions of Parkinson's disease and Huntington's disease, where Parkinson's disease is going to be this interruption of nigrostriatal dopamine into the caudate and putamen, whereas Huntington's disease is a deficit of GABAergic output coming out of the neostriatum to the pars reticulata of the substantia nigra. Then the other interesting thing is to look at the subthalamic nucleus and the globus pallidus, where you can see the globus pallidus zona externa is going to the subthalamic nucleus, and then the subthalamic nucleus, in addition to the input coming in from the neostriatum, influences the globus pallidus zona interna. Now, what's so important about the subthalamic nucleus? Again, we can look at this in terms of neurological dysfunction, the neurological dysfunction that we see with Parkinson's disease. So back about, I guess now, almost 15 to 20 years ago, I can't remember exactly the year, but the Nobel Prize for Physiology and Medicine was awarded among 3 different labs, the heads of the labs, for 3 very different things. One was Paul Greengard, of course, who was instrumental in understanding all of the cascades of G protein coupled receptors and how receptors basically work and whatever in synaptic function. The second, of course, was with Eric Kandel for a cellular basis of learning. And then the third was given to Arvid Carlson. And Arvid Carlson, in his early work, was the person who basically figured out that with Parkinson's disease, with this loss of nigrostriatal input into the caudate and putamen, ending up with people with Parkinsonian tremors and things like that, that you basically do what is called L-DOPA treatment. That is, you give the precursor of dopamine to a patient, and that whatever remaining dopamine cells could basically use that, and it could ameliorate the effect. But what we find out with L-DOPA treatment, as Parkinson's disease progresses, it becomes less and less useful. So the whole thing of L-DOPA treatment is a simple replacement of the caudate and putamen. But then that gets lost. So now other folks went down, neurologists and neurosurgeons went down to a further point, and that's the subthalamic nucleus. So if you now place an electrode into the subthalamic nucleus and now apply electrical stimulation, or that the patient can apply electrical stimulation to the subthalamic nucleus, what are you doing? You're bypassing the whole basal ganglia circuit to allow the person some release from movement. Because what basically happens in late Parkinson's disease is not only that you're now beyond tremor, and you're literally almost in a catatonic frozen state. And that becomes incredibly painful, et cetera, et cetera. And the subthalamic nucleus, it's not like a substitute to make a nice, easy movement, but to make some movement for some relief so the person can amble around a bit and not be subject to the chronic pain of all of the joints sitting in one place at once. Okay, so there is this circuit, and again, depending on how you're looking at things, you're seeing the importance of these things. So very, very, very simply, the basal ganglia and the three components of it have a very simple interconnection with one another. The caudate and the putamen send their projections. They don't send their projections anywhere else. They're sending their projections to the globus pallidus. It's a unidirectional output. And as far as the caudate and putamen is concerned, they don't talk to the rest of the brain. They only talk to the globus pallidus. And what's their talk? Their talk is primarily inhibitory. So it is you're going to see an enormous number of GABAergic outputs coming from the caudate, coming from the putamen, and synapsing on the globus pallidus. And they're basically telling the globus pallidus, whenever they're activated, to stop, okay? And then what you have are different parts of the caudate and putamen telling different parts of the globus pallidus what to do. So now the globus pallidus is sort of dumb, other than it gets subthalamic inputs, it gets some other kinds of inputs, but most of its outputs are being restrained in some way by the caudate and putamen. And there are two major forms of globus pallidus outputs. One is called the fields of Farrell. The second are called pallidofugal outputs. Fugal means away, so they are efferents leaving the globus pallidus. So always, I love this. Yeah, we've come across Rolando, we've come across Sylvius, we've come across earlier today Ammon, people who have named and left the brain named in here. So here what we have are the fields of Farrell. Okay, so Farrell was a French neuroanatomist who studied some globus pallidus outputs. Okay, so what I'm going to basically do is a psychoanalytic view of Farrell. What I'm basically going to do is I'm going to call him a narcissist. Why do I call Farrell a narcissist? The three structures that he independently identified as coming out of the globus pallidus was the ansa lenticularis. Remember, lenticular nuclei are putamen and globus pallidus, the lenticular fasciculus, and then the thalamic fasciculus. So he identifies three different globus pallidus efferents. And what he does is he calls each one of these three different efferents a different name. He gives the ansa lenticularis the H field of Farrell, the thalamic fasciculus the H1 field of Farrell, and the lenticular fasciculus the H2 field of Farrell. Now, why do I call him a narcissist? Well, his first name was Henri. Okay, so we have Henri Farrell naming the H field of Farrell, the H1 field of Farrell, and the H2 field of Farrell. Now, where are the fields of Farrell, or where are these fiber tracts passing through? Well, if you remember, we talked about the hypothalamus. And what we did was we looked at the hypothalamus. We saw the medial border, of course, was the third ventricle. The ventral border, of course, is the bottom of the brain. Then the lateral border was the optic tract in the internal capsule. But then the dorsal border of the hypothalamus was an area called the zona incerta, INCERTA. And this is the area through which the H1, and the H2 fields of Ferrel, these Globus Pallidus efferents, run. And it is also an area that contains dopamine-containing cells put through it, the so-called A11 cell group. The A10 cell group was the ventral tegmental area. The A9 cell group was the substantia nigra. The A11 cell group is found up here in the zona incerta. Now, what are all three of those H1 and H2 fields of Ferrel going? It is the Globus Pallidus efferents that are running back into the thalamus. So it is the striatal output going back into the thalamus, specifically what two nuclei of the thalamus? The VA nucleus and the VL nucleus of the thalamus. So they are projecting to the VA and VL thalamus. But there are also 4 different pallidofugal outputs. So first of all, the Globus Pallidus also projects to the substantia nigra pars reticulata. It then projects to the subthalamic nucleus. It then projects to the red nucleus, and then it projects to the VTA and the colliculi. Give me a second here. I'm going to look at, okay, I'm just looking at what the time is. We're still going strong here. Okay. Okay, so here we are with these outputs. So what we can now start to think about is different types of important structures. So one thing that we basically know about is that some parts of the somatosensory cortex and some parts of the motor cortex and some parts of the premotor cortex all will send out outputs into the putamen, and consider for this also the caudate. Okay? The caudate in turn is also getting inputs from the substantia nigra pars compacta. So you're having that output. Then you're getting output from the centromedial nucleus of the thalamus into the putamen. So in the caudate and putamen, you're getting enormous types of glutamatergic output coming out of all of these cortical areas, the supplementary cortical areas, the diffuse cortex, and motor cortex and somatosensory cortex. Then and it's getting dopaminergic inputs and thalamic input. Then what basically happens is the putamen is either going to provide input into the external area of the globus pallidus or the zona interna of the globus pallidus. It also is going to provide strionigral GABAergic input into the substantia nigra pars compacta. So now here we are in the globus pallidus. The globus pallidus zona externa has reciprocal connections with the subthalamic nucleus, whereas the interna of the globus pallidus is now through the fields of Forel going to the ventrolateral nucleus, the ventral anterior nucleus, and other thalamic nuclei. So what you can basically see with this globus pallidus output is both pallidofugal that's basically going to be there to precipitate or not precipitate some type of motor action, or it's going to be providing further feedback in that striatothalamic cortical loop that keeps going round and round until something gets off. So now, the other interesting aspect about this is to basically look at the activity of the basal ganglia when we want to look at this from a point of view of excitatory input, which is glutamatergic primarily, GABAergic input, which is primarily inhibitory, and then we're then going to be looking at encephalinergic input, and then we're going to be looking at two different forms of dopamine input. And the two different forms of dopamine input can either be excitatory or it can be inhibitory. And that's what we're going to be basically looking at when we see this. So now how can you make dopamine input excitatory or inhibitory if you have the same damn molecule? It's how they interact with receptor superfamilies. There is 5 major dopamine receptors: D1, D2, D3, D4, and D5. However, they're divided into 2 different superfamilies: the D1, D5, and then the D2, D3, D4 superfamily. The D1, D5, or overall D1 superfamily is going to be excitatory. Whereas, so when dopamine binds to that receptor, the cell upon which that receptor resides is going to have an EPSP. And that's the dopamine D1 superfamily. If there's a D2 superfamily type of receptor, and it is on a dopamine cell, and sometimes you can be on exactly the same cell in the striatum, in the caudate and putamen, that contains both D1 and D2 receptors, that D2 receptor is going to produce an IPSP. So now you have to ask where on the putamen or caudate cell you are. Are you on a dendrite, or are you on a cell body very close to the axon hillock? And very often what we find is, even though there might be fewer inhibitory receptors, but if they're all found very close to the axon hillock, their inhibitory effect has a much greater effect than some dendritic synapse, okay? So you have to basically study the sub-architecture of these neostriatal cells. So what we can basically see here, if we want to start out from the thalamus, and we basically look at sensory input coming in. Basically, what you see here is largely a glutamatergic input into the cortex. And the cortex, in turn, will either excite the neostriatum, caudate, and putamen, or they will go all the way down and do a excitation through glutamate of the subthalamic nucleus. So basically, think about a somatosensory input, then some motor type of response, and it's all very simply excitatory. But now, what do we have? In the putamen, what you can basically have is if the cortex starts to fire and say, oh, we got some very important input, and it's either a motor output or a sensory output and coming, it excites the putamen. And it can excite the putamen in two different ways, through an indirect pathway, or it can excite the putamen through a direct pathway. Notice that both the indirect pathway and the direct pathway are both inhibitory. They're basically going to be telling the Globus Pallidus zona externa and the Globus Pallidus zona interna to stop. But why is it doing that? Because what the Globus Pallidus zona externa does is produce inhibition. So what you're basically doing is if the thalamus is exciting the cortex and the cortex is exciting the putamen is now inhibiting the Globus Pallidus zona interna and externa to do what? To stop inhibition. Because what the Globus Pallidus, if there was no putamen, if there was no cortex, if there was only just the Globus Pallidus, we would have a creature who would always be in a state of what? Stopping, not doing anything, because what is the major output of the Globus Pallidus? GABAergic inhibition of the subthalamic nucleus that then allows behavioral output, or of the Globus Pallidus zona interna with GABAergic input on various parts of the thalamus and the pedunculopontine nuclei that then activate the reticular formation. So there is the Globus Pallidus is, just in that immortal words of Meatloaf, the female protagonist of Meatloaf in Paradise by the Dashboard Light, stop right there, okay? Stop. So what is basically happening, whenever the putamen is working and is being activated, it is inhibiting the Globus Pallidus and thereby disinhibiting everything that's downstream from it. It's allowing movement to happen. Now, on top of that, so we have that in terms of what? Very short-term things, because we're getting sensory events coming into the thalamus, we're getting cortical events, making decisions, telling the putamen to work, so that we can have a behavior. But underlying all of this is some system that basically puts us in some degree of readiness. So we're sitting here and we're doing things, and if we are writing frantically, or I don't know, doing whatever the hell you're doing because you're all blacked out, what you basically have is this sort of resting state that is modulated by the substantia nigra pars compacta. And here is where this dopamine input comes in. So now this dopamine input coming from the substantia nigra pars compacta can act on D1 receptors. And when it acts on D1 receptors, what basically happens, there is in the striatum, there is an EPSPs. And those EPSPs can then release, look at what it can release. It can release substance P, it can release GABA, and whatever. Now, on the other hand, if we now bind to D2 receptors, what do we now do in this pathway? We inhibit this pathway. The D2, binding to D2 receptors blocks what? Glutamatergic, it blocks GABAergic output, it blocks encephalinergic output to the globus pallidus zona externa. So, when we are here, I want to get back one. With the direct pathway, even though it's you're exciting something that is going to inhibit the globus pallidus zona interna. So what you're doing here is increasing the inhibition. Whereas when dopamine is working through D2 receptors through the indirect pathway, you're inhibiting this, and you're inhibiting an inhibitory pathway, which now does what? It now makes the globus pallidus zona externa more having a greater type of effect. So what we can see here is that this is the underlying etiology of the D1 and D2 receptors, and then how those D1 and D2 receptors are affected differentially by the slow progressive loss of brain dopamine in Parkinson's disease may then exacerbate that imbalance that much further. So I think what I just did there is, yep, it's exactly noon. So what we can do at this point is we're going to stop at this point, and then we're just going to, at the beginning of the next class, literally review this striatal circuit and review it in terms of the thalamus and the cortex, and then review it in terms of various types of outputs and extrapyramidal motor behavior. We will then, in the next class, which is the class before Thanksgiving, switch gears. We will then start to go into the brain in section, looking at the coronal sections. The next week after that, we have Thanksgiving, and I hope you're not here for that. And then the week after that, we will continue the coronal sections and the sagittal and horizontal sections. And then in the final week, we will cover that extra class that we're doing on the 10th of December. We will cover the cortex and a quick review of neurochemical neuroanatomy. So go through this. And what I encourage you to basically do is start to think about some type of response. You're going to move your arm or something. And then at each point in the circuit, ask what stops you from moving your arm, which keeps your arm in place, and what will eventually activate it. And you're basically seeing this interplay between excitatory types of inputs and inhibitory types of inputs, and then the very powerful role of disinhibition. Disinhibition is not just pure excitation. It results in the same kind of thing, because if you inhibit an inhibitor, you allow excitation to occur, but you allow excitation to occur through some type of very clear kind of funnel. Okay, So I have just a couple of questions here, and maybe none of you can answer me this, but I did notice over in the last couple of weeks that when you guys took the midterm, there were 31 of you. And I've noticed like today, there was a max of about 25. Last week, there were 25 to 26. Are people hearing any kinds of input that for some reason the other people can't make it? Or are they asking for the recordings? Or they just give up the ghost? I don't know. Anybody have any input on that?
Professor Bodnar, I know I missed 1 week because my internet was out.
Oh,
So,
gee.
like stuff
Okay.
like that happens.
So shit happens. All right. That
Yeah,
no.
basically.
And I'm hoping you were able to get the recording.
Yeah, I could see all the recordings. It's nice
So
having
one
those
of
as a
the
backup.
things I'm asking people to do, and even the people who aren't here today, and if they're still active in the class and they're still using the recording, please, people, if people have questions, whenever you're recording or whatever, or you just have questions afterwards, always ask me. I can always answer your email. And very often, if you ask an individual question, and I think it's a question that can be clarified to everybody, I'll send everybody an email with the answer, okay? So another thing to look forward to, I know I'm taking a little extra time here, but one of the things that you'll look forward to is the Thursday after Thanksgiving, that one around the 3rd of December. What I'm also going to be providing you is the magic questions. There's as you know, I love popular culture, and one of one fairly modern playwright, although he's pretty old by now, is a guy by the name of David Mamet. And he wrote the magnificent play American Buffalo that Al Pacino always seems to go into and see. But then he made what I found was one of the great plays of the 1970s and 80s, which is called Glengarry Glen Ross. And it's a bunch of guys selling shit real estate in Florida, like in the Everglades or in the desert in Arizona, to retirees and everything, and trying to get money. And the point of the matter is they're a little branch office up in the Bronx, and then they have the main office down in Manhattan. And in a classic role in a movie that they made of this was Alec Baldwin, who obviously today plays Donald Trump on Saturday Night Live. He came in as the guy from downtown. He doesn't even have a name. And he's holding up a bunch of little 3x5 cards. And he goes, here's basically, it's a card filled with names of people who are possible suckers, right? And he's holding it and saying, you can have these cards. So you want to have the names so you can make the sales and everything like that. So on December 3rd, I'm going to give you the new set of cards. On the cards are going to be a sort of supersized set of questions that could be on the final exam. The final exam will come out of these questions, but there's more questions than what are on the final exam. But it'll give you a head start to start preparing and looking for that part 2 of the final exam. And that'll happen on December the 3rd. Okay? So here we have moved through. And again, the important thing here that I try to do is, like when I talked about the thalamus, I talked about the limbic system, or I talked about the hypothalamus. Here I'm talking about the basal ganglia. Sometimes we have to put these pieces, individual pieces of the puzzle back together. And if you find that you're having a real trouble doing that and whatever, please ask me questions, okay? So other than that, have a very good week. So have a good week, gang. Bye.
Bye.